Good morning. Welcome to the 2021 American Oil Chemist Society annual meeting. My name is Scott Bloomer. I work for AOCS. I'm the Director of Technical Services, and today it is my joy to be the host of this session, which is an ePoster pitch competition for biotechnology. We have some very strong scientists as judges today. I would like to introduce them first. Doug Hayes is a professor at the University of Tennessee in the USA. Doug and I go back a long ways. We've known each other for many years. He has served on the governing board of AOCS and is now the editor-in-chief of one of our flagship journals. We also have Susana Ferreira-Dias, who is a professor at the University of Lisbon in Portugal, another very strong scientist. I don't know her as well, but I have heard of her throughout my career. And so we're very privileged to have her. And we have Sarah Willett from industry. She works for Kerry. So obviously she has a very strong background and works in research and development. So thank you very much for these judges who are going to be providing their feedback today. The format today will be that there will be 3 student presenters, each of which will have 5 minutes to present. And as the audience, you're able to enter your questions for these speakers in a Google spreadsheet, which has been made available. After the close of the person's talk, each person will be given a chance to ask questions. The judges will ask questions and I will ask questions from the Google spreadsheet. And then after all of the presenters have taken their turn, there will be about a 3 or 4 minute time window where the audience is asked to vote. So there will be a vote button that will show up and you will be able to vote, and our judges will vote, and each judge's vote will count for 1 and the audience vote will count for 1. So there'll be a total of 4 judgments that will be entered. At the close, we will announce a winner and a runner-up or a second place. And everybody that participates will get a certificate and a free membership next year in AOCS. And also you'll be mentioned in our newsletter. So I would like to start with introducing our first candidate here. We have 3 presentations today. And the first one is Ahmad Alhomodi from South Dakota University here in the USA. The title of his talk is Applications of Fungal Mycelium, Co-Cultures of Fungal Mycelium during Solid State Fermentation of Canola Meal for Potential Feed Application. I'm very excited to hear this talk because canola meal is a vastly underutilized substance and there's a lot of it. Ahmad is currently pursuing a PhD and he is focused strongly on canola meal extraction. Ahmad, would you like to start?
Good morning, everyone. First, I would like to thank all of you for attending my presentation, which is under the title Application for Co-Culture of Fungal Mycelium during Solid-State Fermentation of Canola Meal for Potential Feed Application. Canola is an important oilseed crop, which is ranked second after soybean. When canola seed is a process for oil extraction, 2 fractions are generated. We have oil fraction and meal fraction. And it is estimated that for each 1 kilogram of oil produced, a 2 kilogram of meal generated. And oil has a different industrial applications such as human food and biodiesel, while meal is used in animal diet. If you look at the canola meal benefits, even though the meal is rich in protein with well-balanced amino acids, its inclusion in animal diet is limited due to the presence of anti-nutrition factors such as glucosinolate, phytic acid, high fiber, and oligosaccharide, beside low protein digestibility. So the bioprocessing goal is to enhance the nutritional and the commercial value of canola meal by increasing the desirable components such as protein and decreasing the undesirable components such as anti-nutritional factors. If you look at the possible solution, several attempts have been done to improve the canola meal, such as milk pretreatment, microbial fermentation, pretreatment followed by microbial fermentation. All these processes showed the potential in decreasing the anti-nutrition factor, but fibers is still a challenge. The drawback of using tail end dehoning process is the high energy input requirement beside the loss of protein in the whole fraction. Thus, our project is to exploit the synergistic relationship among three different fungal strains, Eurobasidium bollenans, Neospora crassa, and Trichoderma ricei, in co-culture incubation under solid state fermentation to improve the canola meal composition. In our material methodology, we started with a canola meal, or 50% of canola meal on dry basis. The meal was autoclaved and then inoculated with the mono and co-culture of three different fungal strains, Eurobasidium bollenans, Neospora crassa, and Trichoderma ricei, beside an inoculated control. All the flasks were statistically incubated at 30 Celsius for 168 hours, and at the end of fermentation, all the samples were dried, grind, and analyzed for anti-nutrition factor and crude protein. Here in our obtained result, before I start talking about my result, let me tell you what the abbreviation are. If you look at the first row, we have TSR, which is a total solid recovery, CP, crude protein, TC, total carbohydrate, CF, crude fiber, ADF, acid digestion fiber, NDF, neutral digestion fiber, GLC, glucosinolate, PA, phytic acid. If you looked at the first column on the left, we have HECM, which is hexane extracted canola meal. And at the bottom, we have AP, which is apollolans, NC, encrasa, TR, Trichoderma ricei. So in our result for the monoculture, encrasa showed the least total solid recovery, and it was the best in increasing the protein content and reducing the crude fiber, acid digestion fiber, and neutral digestion fiber. Whereas the apollolans was the best in reducing the total carbohydrate and the glucosinolate, but TRCI was the best in reducing the phytic acid compared to the other fungi. In the co-culture fermentation, the combination of apollolans and encrasa showed the least total solid recovery, and they were the best in increasing the protein, reducing the total carbohydrate, reducing the crude fiber, reducing the acid digestion fiber and neutral digestion fiber, whereas apollolans with TRCI were able to reduce the glucosinolate and phytic acid. So in summary, all the strain individually and co-culture increased the protein compared to the raw meal. The combination of apollolans and encrasa were the most effective in reducing the fiber content, and the co-culture of apollolans with TRCI were the most effective in reducing the glucosinolate. So in conclusion, the co-culture and fermentation improved the canola meal composition, but that improvement is different based on the fungal combination. So the selection of microbe or the fungi need to be based on the goal of the final product. And finally, this improvement in canola meal could help in solubilize the protein and increase the protein extractability, which is considered a big problem in the protein extractability that we are currently studying. That leads me to the end of my presentation. Thank you for your attention, and I will be pleased to take your question.
Thank you, Ahmad. That was very interesting. So now we have a question in the chat, and we have our judges here. Judges, do you have a question to start with?
Yes.
Professor Ferrari, yes?
Thank you, Ahmad. It was a very nice presentation. Congratulations for your work. Very interesting. But I have a question because you tried the strains isolated and 2 by 2, and also all the 3 together. But in almost, I think, for all the parameters you followed, when you use the 3 strains together, the results were worse than using only biculture. Can you have an explanation for this, or not yet?
Yeah, that is a very good question. It's based on, because when I use two microbes together, sometimes the product of one or these 2 microbes could inhibit the other microbe, for example. So for the TRCI is known for the production of cellulotic enzymes, but the presence of many like a sugar and the soluble sugar in the media cause a feedback inhibition. That's why you see that there's no a good result when they are together, because if there's a lot of sugar, for example, it's because of feedback inhibition for this microbe. There was thus the fermentation is not like when there's just 2 microorganisms.
Okay, thank you. Thank you.
I'd like to ask a question. So I'm Doug Hayes. Thank you, Ahmad, first of all, for giving the presentation, and very good work, very interesting. Just a short question. Have you looked at any process economics, or is that something that you're going to be looking at later on in your dissertation work?
Yep, that's a great question. We haven't looked at that yet, but I mean, what we are using is not that, costly. And I think in the future, we have to look at that when we want to scale up and see how much that could be cost and what is the process cost. Yeah, but currently we haven't done that yet.
Yeah, actually, it's a more difficult question than anybody ever tells you about when you're in graduate school. You have to get engineers involved, and generally one of the interesting things to think about is you really want to adapt some other process that's already been costed out, so people know what it's going to cost, and that would be very. I'm a very big believer in canola meal utilization, so this is exciting. We have an opportunity for new enriched products this way. One question I was wondering about was, how did you select your co-culture strains? Obviously, there are millions to choose from.
Yeah, all the fungi that we have are generally recognized as fungi. And we have a mini collection, and we choose these microorganisms based on their enzymatic ability, based on the previous studies, to see if and also we did apply a dome culture to test any antagonistic behavior among these microorganisms before we use them. And we found that they grow together without any antagonistic behavior. And we saw that their ability in producing a cocktail of enzymes, that's why they were selected based on that. Yeah.
Excellent, thank you. Well, I don't see any other questions unless any of the other judges have a question. Thank you very much, Ahmad, for a very interesting talk, and I wish you great success in your future.
Thank you very much.
I'm sure you'll tangle with many interesting problems.
Thank you very much. Okay. Thank you.
We have our next speaker, Aurore. Would you mind turning on your webcam? And Ahmad, you can turn yours off. There she is. Aurore Cournoyer is at Laval University in Canada, and she's going to speak on impacts of discoloration on the antibacterial and antifungal bioactivities of a porcine Cruor hydrolysate. And I must confess, Aurora, I had never thought about the question of Cruor and the fact that it's a rich raw material and a source for a lot of potential products. So you started in September 2020 at Laval, and so you are just really launching into the hard work of it now. So I'm very excited to hear your paper. If you wouldn't mind turning off or turning on your microphone, and I will be glad to hear what you have to say. Thank you.
Okay. So hello everyone. So I'm going to present you today a part of my PhD thesis, which focuses on the in vitro antibacterial and antifungal bioactivities of a porcine Cruor hydrolysate. So first, it's important to know that the pork industry is one of the main sectors in the agri-food field in Quebec. They provide about 25 to 30% of the Canadian consumption of pork. Nevertheless, consumers' concerns regarding this sector are increasing. They particularly care about the pollution, the production of waste, and the use of synthetic preservatives. And slaughterhouses produce many byproducts such as blood, which represent 30 million liters per year in Quebec. So you have to know that the blood can be separated in two parts, the plasma and the Cruor. And the Cruor is the red cells part. And it contains about 90% of hemoglobin. And so thanks to its abundance in protein, it's an ideal substrate for proteolysis. And it had been demonstrated with the bovine hemoglobin that after enzymatic hydrolysis, you can produce the bioactive peptides, specifically antimicrobial peptides. And these peptides can be used within the pork industry, which creates a circular economy. But there is a lack of knowledge concerning the bioactive peptides from the porcine Cruor hydrolysate. And moreover, it's important to know that the color and the metallic flavor of M that is contained in the hemoglobin can limit its commercial potential. So that's why the main objective of this study is to evaluate the impact of the discoloration of a porcine Cruor hydrolysate on the antimicrobial activities and on the peptide profiles that are generated. So concerning the method, first we had to produce the Cruor using centrifugation, and then we did an hydrolysis with pepsine using these parameters, and a discoloration using acidification. And then we evaluated the antimicrobial activities using a qualitative method so based on an agar diffusion, and a quantitative method which determined the minimum inhibitory concentration. So we did these both methods on bacteria, yeast, and molds. And then we also determined the peptide profiles with UPLC coupled with MSMS system, which allow to separate, identify, and characterize the active peptides. So concerning the results, we can easily see on this overlay of the raw and discolored hydrolysate spectra that there are differences between the both hydrolysates, and mainly at the end of the spectra between 20 and 27 minutes of retention time. And also you can see that no peak of M was observed on the discolored hydrolysate spectrum, which shows the effectiveness of the discolored step. So we identified the lost peptides, and then it was relevant to see whether these lost peptides had an impact on the bioactivities or not. And as you can see, no inhibition was observed on bacteria, but antifungal activities was observed for both hydrolysates, so the raw and the discolored hydrolysates. And we observed a significant differences between our two hydrolysates, So the raw hydrolysate was, for example, for the first strain, which is Saccharomyces boulardii, you can see that the raw hydrolysate is 10 times more efficient. So the strain is more sensitive to this hydrolysate and less sensitive to the one discolored. So following these results, we were able to demonstrate for the first time that porcine Cruor hydrolysate have an antifungal activity, and that the discoloration causes the complete loss or the diminution of some specific peptides that have an impact on the resulting activity of the hydrolysate. So the general perspectives of this project would be to produce a new and bio-based ingredient. And also we decided to synthesize some of the lost peptides to see their activity purified, and the results are yet very encouraging. So thank you for listening, and I'll be happy to answer your questions.
Thank you, Aurora. That was very concise, and it's obvious you've gotten a great start. So I appreciate your perspective.
Yeah, thank you. Thank you very much.
Susanna, did you have a question? I see you're unmuted.
Yeah, thank you. Congratulations, Aurore. It's a very, very nice presentation. And I know nothing about your subject, but with your presentation, it was much clearer for me now. But I have some questions that are more curiosity, because I don't work in your field. And you said that these peptides can be used as food preservatives. Are they already used in the food industry, or is it new? It's our idea to use them in the future? And do you think, just two questions in one, and what do you think about discoloration of your raw material, because you observe the loss of activity? What do you think about all the process in terms of use in food technology, for instance?
Yeah, so they are not yet used in the industry, but there are several studies that show that they can have very good activities in situ. They tested some peptides on the meat, and it shows a very good result. So it has the potential to be used as preservatives. And so it's not yet used, but it's a project, and it's very interesting because it could create a circular economy, and I think it's very interesting. But yeah, of course, the discoloration, it's adding a new step. So it would be better not to add this step, but because of the color, if you want to add this hydrolysate on light meat or something like that, it could create an issue. So that's why we try to discolor to see if it has an impact or not. And also for later, we will try to fractionate the hydrolysate with some techniques that use membranes, and the M can be a problem. So that's why we really wanted to, at first, compare the two hydrolysates.
Thank you.
I don't know if it's answering your question.
Scott, if I may ask the next question.
Please, Professor.
All right. Thank you, sir. Appreciate it. A gentleman and a scholar you are. Scott and I are friends, so we just ignore that. All right, very good presentation. I was very interested because I'm working with antimicrobial peptides that actually attack bacteria. They're designed to, they penetrate the biomembranes and kind of open up pores. I was curious, what is the underlying mechanism, or do you know this, for the products that you're producing and how they may be affecting yeast and fungi and causing that antimicrobial activity.
But the antifungal mechanism are quite close to the antibacterial mechanism. They also have to penetrate the cell membranes, and I know fungus have another membrane that bacteria doesn't have, but it's quite the same mechanism, and the peptides will penetrate and cause the loss of metabolism, the metabolites, and then the cell will die, like bacteria.
Thank you.
Thank you.
You're welcome.
Very good presentation. In the interest of time, I'm going to skip the remaining questions, but thank you very much for a very interesting and well put together talk and a very good presentation.
Thank you.
So thank you. Tailoring Camelina sativa seed fatty acid composition via fast neutron mutagenesis. So he's treating these seeds and looking at the fatty acid composition thereof. So this could be a very powerful and interesting tool. So I'm looking forward to hearing this talk. Winston, please begin.
Hello everyone, I'm Xinjie. In here, I'm gonna do an introduction of my poster. The topic of my poster is tailoring Camelina sativa fatty acid compositions via fast neutron mutagenesis. And let's begin with the introduction. For Camelina oil, the most amazing part is it contains a very high amount of polyunsaturated fatty acids, such as essential fatty acids like linoleic acid and alpha-linoleic acid, which contain about 20% and 33% of the total oil content, respectively. And Camelina sativa has a hexaploid genome structure. It means Camelina has 3 subgenomes, and this genetic redundancy will reduce the likelihood of lethal mutations. The methods we use in this project to introduce mutations is fast neutron irradiation. And the major mutations caused by this type of mutagenesis approach is deletions. And this kind of mutations have chance to completely knock out genes in Camelina genome. Experiments in this project can be generally divided into 2 parts. The first part is phenotyping, and the second part is genotyping. For phenotyping, the 2 major experiments is germination test and gas chromatography analysis. Here shows the result of germination test. The first group is the control group, and other groups are the treated group. And we can see at 24 hours, higher dosage treatment has less seed germinated. But after 48 hours, all the group has a germination rate close to 100%. And this indicates that fast neutron treatment can slower the germinating speed of the seeds, but fail to lower the germination rate of the seeds. And this chart displays the comparisons of the fatty acids composition of seeds between mutagenize and reference. And here the reference is Midas. And we can see the two mutagenides I selected shown in this chart had a very different content of linoleic acid and alpha-linoleic acid compared to the Midas. And this kind of change of fatty acid compositions is desired, and the mutagenides contain this desired lipid profile will be used for further genotyping analysis. So the second part of the experiment is genotyping. The two major techniques we use is PCR scanning and genotyping by sequencing. For PCR scanning, it's used to detect whether there is deletions occurred into the Camelina genome. And genotyping by sequencing, also known as GBS, is more focusing on understanding what exactly happens inside Camelina's genome. And after this experiments, we also will do backcrossing. This backcrossing is in order to backcross the mutagenides with desired lipid profile back to the commercial Camelina species in order to induce those desired traits in the commercial Camelina species. And this can help to improve the Camelina oil quality. And this will be all the content of my introduction. Thanks for listening.
Thank you, Winston. Very interesting talk, and
Thank you.
very a vast field of potential applications and research. So thank you very much. Sarah, did you have a question?
I did. First of all, I just want to say congratulations, Winston. I thought you did an amazing job presenting your research. I was wondering if you could explain more on your results slide where you compared the Midas control with your two tests. If you could explain more why the test in blue showed higher linoleic content and the test in orange showed higher linolenic content, if there was a reason behind that.
Yes, because for the mutagenides, we treated with fast neutrons irradiations, and after treated with the mutation, the fast neutron irradiations, there could be some mutations introduced into the plant genomes, and if the mutation is occurred in the genes that associated with some kind of enzyme genes, like some genes associated with like fatty acids elongase or fatty acids desaturase, then this mutation may be block these genes expressions or if improve those genes expressions and can lead to this different lipid profile when we do the GC experiment.
Thank you very much. Here's a question. Gandoic acid is a very uncommon fatty acid, and I wondered if you thought about prioritizing it because it has potential for use in cosmetics and as a transfer agent to bring things through the skin. Has that target been, have you been made aware of that potential target for a market for the altered Camelina?
Yeah, thanks for your question first. This is a very good question. Yeah, and for gandoic acid, because this kind of fatty acids is not very reproducible, this guanidinic acid is good for our human healthies. And so for industry, if we the main interest in the market is to have a much more healthier oil products. So we want to improve Camelina oil qualities. We hope to increase like omega-3 and omega-6, this kind of fatty acids content into the oil products instead of other fatty acids. So for guanidinic acid, because they didn't have some reproducible this kind of fatty acids have a healthy effect on our human body. So we hope to eliminate or reduce those accounts in the Camelina oil products.
Okay, thank you very much. Susanna, did you have a question too?
Yes, thank you. Winston, congratulations for your very nice work. And I just would like to ask you, in all these mutants you got till now, did you have already one that maximizes in the yield of oil and fatty acid composition? That means it is the best in terms of oil yield and fatty acid composition, or is it not possible?
Yes, thanks for the question. For the we cannot say we can find the best or the maximum oil product, oil content of the seed, because we don't know what the mutation caused on this plant genomes. So it may show different phenotypes or genotypes after treated with fast neutrons. But so far we have many different phenotypes of these fatty acid compositions, like I showed in the presentation. Some have higher omega-3 or some have lower omega-6, something like that. And there could be some phenotypes like higher omega-9 or oleic acid, or even some seed treated with fast neutron, they can lead to low germination rate or other kind of phenotypes, like different visual phenotype, like the leaf will be a different shape. And also for the oil content, so far we don't find a light have a very high oil content, but the average oil content so far is about like 35% or something around this. And we don't know if we can find some amazing oil content mutanites later or in the future. So it's very hard to say which one is the best, or yeah, because they all have different characteristics.
Thank you. Congratulations.
Thanks.
Scott, may I ask a question?
Please, go ahead, Professor.
Okay, sure. Thank you, Winston, for presenting, and I also congratulate you.
Okay.
I guess, just if you can speak to why you chose fast neutrons to cause mutagenesis versus other methods. I wasn't sure if there was a specific hypothesis that you might be testing in your research work.
Yeah, we choose for this question, because we choose fast neutron radiation mutagenesis method to do this project, is because fast neutron the major mutant type caused by the fast neutron is deletions, and for Camelina's genome structure is hexaploid genome structures, and so which means it has many genetic redundancies in their genomes. So if we can lead to deletions into the genome structure, especially some large deletions, it may have a higher chance to completely block or delete genes, some kind of genes. And compared to other methods like using the chemical mutagens like EMS, those chemical mutagenesis methods, the major mutation types caused by those methods is the single nucleotide substitutions. This kind of mutations is just change one single nucleotide, and this is very hard to determine, and also it may not have a very numerous influence on the phenotype. And so we choose a fast neutron to have a try.
Thank you very much for your answer. May I ask one more question, Mr. Moderator, Dr. Moderator?
Please
I was
do.
curious where you did this work at. Did you use the Neutron facilities at Ottawa, or where did you do this work at? What was your neutron source?
We treated the seeds in another institutions in the McAllen Nuclear Radiation Center at UC Davis in California, not in our center. Yeah.
Thank you.
Thanks.
All right, I think those are all the questions. Thank you very much, Winston, for a great presentation, and I thank you, all of our students. And now we're switching to the voting time. So our judges have received a matrix and they'll be tallying their vote, and the scores will be announced here, and the audience will have a chance to vote. So I will now talk about some other things that are happening in the AOCS annual meeting. And these are all of interest to the Biotechnology division. Of course, there's an extensive program for the Biotechnology division, but I did find a paper that I thought looked very interesting that was in another division, and this was from a monoenzyme. And the title of the talk is called Enzymatic Editing of Vegetable Oils to Obtain a Low Diglyceride Oil by Two Methods: Specific Hydrolysis of DG and Conversion of DG to TG using a unique lipase. So I've always been interested in new lipases that are popping up and their potential. So this is by Tomohiro Kimura at Amano Enzyme in Japan. Unfortunately, I don't have the time noted here. In addition, today at 4 o'clock, there will be a session for the Biotechnology division called Gene Editing Technology and Advancements in Agriculture. So that's one of the talks we just heard was on gene editing. And Wednesday, tomorrow morning at 7 a.m. Chicago time, there will be a session called Emerging Biotechnological Developments in Lipids. This kind of would be the throwback to the kind of sessions we used to have 20 years ago in the Biotechnology division. Also tomorrow at 4 p.m., the breeding and technology for improving content, nutritional quality, and functionality of plant proteins. So this is a co-session with the protein and co-products. Judges, have you entered your votes? Okay, good. In addition, Thursday at 1 p.m., the Biotechnology division will hold a session called Plant and Algae Lipid Biotechnology and Genomics. Friday, there'll be a session on renewable or biorenewable polymers, which will be at 1 o'clock. And then next Monday, there'll be a session at 1 o'clock called Functional Lipids Biocatalysis 1. And that, of course, biocatalysis has been the name of a session for 100 years in the Biotechnology division. Next Tuesday at 1 o'clock, there'll be a session on bio-based surfactants, and it looks like Professor Hayes is going to be one of the co-chairs. So I just don't know how you do it all. It just baffles me, Doug.
Good co-chairs.
Good co-chairs.
Just like good moderators.
That's a good secret. Okay, two more quickly here before we enter the votes. There will be a Biocatalysis 2 session next Wednesday at 4 o'clock. And next Friday, there will be a general biotechnology session. And with that, I will ask our organizer to step in. very much for participating today. Thank you very much. And we have received our tally for our votes here. And I would like to, before I announce that, I would like to give a special thanks to our judges today, who are very, very busy people and have taken time from their schedule and really had some input into these young scientists' lives. And that, in the end, is really what's important, is bringing up the next generation. So thank you very much. So our second place winner is Aurore. So congratulations on her paper on the porcine Cruor. And our first place winner was Ahmad, so on the canola meal. And so thank you very much for your participation. And AOCS will, by some mechanism unknown to me, communicate with you about the next steps. So thank you so much for your participation and congratulations on 3 very strong talks. And I look forward to future presentations by you in other AOCS meetings.
Thank you.
Thank you.
Thank you.